In previous lectures, I've already covered the basics of transcription and translation. So the process of gene expression, which is where we have DNA containing the code. The DNA is copied into RNA. So if this is our DNA here, we have what we call a coding strand and a template strand. Now, when the process of transcription occurs, an RNA polymerase here is going to read the template strand, and then whatever that sequence is, it's going to copy it, but using complementary base pairs. So, for example, if this sequence was A, T, G, C, C, A, and so on, as we make this new strand, this will be a U, A, C, G, G, U, and so on, because this is going to be RNA. Right. So that's the overall concept, and you should already know that. Then there's the process of translation, which is where we get all the different types of RNA, which you need to know: messenger, ribosomal transfer, how they're modified, how they all come together in order to then manufacture a protein from that gene. So what we're going to go over now is for bacteria, we're going to add in some levels of detail. We're going to add in some specific or unique things about bacteria that we don't see in just regular eukaryotic cell replication. And we're going to look at the overall process of regulation. So if we have information coded for in the DNA, is it all expressed all the time? The answer is no, it's not typically going to be, and it's not going to be in bacterial cells either. So we're going to go over the terminology about the genes that are expressed all the time, the ones that are not, the proteins and molecules that regulate them, how they turn them on or turn them off, and a couple of other details. So one thing we'll talk about here just briefly before we get into some of these other things has to do with this process of transcription. Okay, so transcription is that process where DNA, is copied into a piece of RNA. And so, it's not, the DNA is double-stranded, both strands aren't copied here. Now, remember, this would be, this coding strand would be T, A, C, G, G, T. So it would look like what we make. So that's the one that actually contains the code, but the one that we are going to be working with is the one that's called the template strand, right? Because we have complementary base pairing. Again, a term that you should be familiar with. If you're not, you need to go back and review that in other lectures. So this particular process, we have the RNA polymerase, which is the primary enzyme doing this, and we find it in bacteria and eukaryotic cells and so on. Before that process begins, so what we're going to have now is, for this template strand, this is the region here that's going to contain the code. The one that we're actually going to read later on. All right. Now we have the RNA polymerase binding to it. The RNA polymerase typically binds to the region of the DNA called the promoter. So a promoter is that region in the DNA. Now what we can have, in addition to that, is a region here, also before that code, called an operator. This is a region where certain repressor proteins can bind. And the repressor protein may or may not bind there. So you may have a repressor protein, attached to this operator, and that means that when the RNA polymerase comes in to bind to the promoter, this will be our RNA polymerase. Either it can't bind or it can't work, depending on what's going on there. So it'll stop or shut down the ability of that gene to be read. it's possible. That's not, sometimes that's going to happen. We're going to talk about the details of that. All right. First off, we'll look at just the polymerase binding. In bacteria, we're going to add a little bit to this here. So this is in addition to things you already know. There's going to be something called a sigma factor. That will bind first. So a sigma factor is a bacterial transcription factor. And transcription factors are proteins, so they're essentially DNA binding proteins. They belong to a number of different classes based on their shape. So there's a class called the leucine zippers that have a particular shape and include leucine. There are zinc finger transcription factors because they use zinc, and the way in which they bind to the DNA that gives them this unique characteristic. All right, and so they're different classes based on their overall structure, but they're all unique, different unique proteins for unique promoters for unique genes. Okay, so, and they're told to bind to the protein by a cell signaling sequence that is telling the cell to express that particular gene at a particular time. So there's a whole cellular communication process going on, which is beyond this, but we're just looking at the idea of what's actually happening at this particular level to express a particular gene. So the cell's essentially saying, I need a particular enzyme, for example, right now. And so a signal would be sent to then read the gene that codes for that enzyme, to make the RNA that codes for that protein, and then eventually to read that RNA and use it to build the protein. That's the gene expression process. So it's a lot of things going on. So there's levels of control for that process. And having a sigma factor bind to the promoter first, so we got the sigma factor, that also provides a level of control. So if we don't have the sigma factor, or if we block the sigma factor from its ability to bind, then we can't bind the DNA polymerase, and then the process can't occur. So we have certain genes that are called constitutive genes. So genes, I'll just... Constitutive. These are ones that are typically always on. Meaning, in this operator region, there's no repressor protein here. And sigma factors can bind, the RNA polymerase can bind, and they can read that gene. These are genes that are also sometimes called housekeeping genes. Because these are genes that are just necessary for the cell to stay alive pretty much all the time and just maintain its basic processes. So the basic processes the cell requires, membrane transport processes, metabolic pathways, things that just have to be running all the time for the cell, these are going to be part of the constitutive genes. So we're not typically turning them on because they need to be always on. And we're not turning them off because if we do, the cell will probably die if we aren't expressing these particular proteins. So those are not ones we're particularly talking about right now. We're just saying they can be regulated often, sometimes to being sped up or slowed down, but they're typically going to be always on. We have two other classes then that are involved in regulation. Okay. We have repressible genes. And we have inducible genes. So repressible are now not always on, but usually. Okay, so they're usually on, but they can be shut down. So these are genes that if they are shut down, the cell's not going to die. It's just going to change what it's doing at that particular time. So they're a code that's coding for an RNA that's going to code for a particular protein that the cell is using for something. But at a certain point in time, we can get, because it's repressible, that means there will be a repressor protein. And that repressor protein can bind then to specifically the operator. So these are the things that you need to know. A repressor protein binds to a region of the DNA called the operator, and that will then prevent the RNA polymerase from being able to read it. That type of gene is called a repressible gene, one that can be shut off if the cell wants it to or needs it to at a certain point in time. Now, in contrast, an inducible gene is usually off, right, but it can be turned on. Can be turned on at some point in time. So with an inducible gene, the repressor protein is just there. It's almost always there. And so we have the genetic information that codes for a particular protein, but the cell isn't making that protein because the repressor is stopping the machinery, the RNA polymerase, from being able to read it. What will happen is usually some type of signal molecule will be required. And that signal would then bind to the repressor protein. And that signal can then remove the repressor protein. And then once it's removed, then sigma factors RNA polymerase will bind, and then it'll be able to read the region of that gene to express it and make the piece of RNA that it needs. Okay, so the main terms here right now that are sort of new terms for you would be a constitutive gene. Genes that are housekeeping genes, we need them all the time. They're never turned off. And then we have genes that are regulated. Genes that are usually on, but they could be turned off, and the cell still won't die. It changes the cell. They're repressible, and they involve a repressor protein binding to the operator region of the DNA. We have inducible genes, genes that are almost always off. We have specific proteins binding to the operator there, and then we need a signal molecule created by the cell that will remove the repressor protein and then open up that gene. Okay, so that's kind of the first part of this, is getting down some of the terms. We're going to have a couple other things go on. Some genes need to be regulated in a different way. So often between the operator and the operator and the promoter region, so again, these are regions of DNA, you might have an untranslated region. So UTR, untranslated region, or untranscribed region, we'll say. So it's a region that doesn't code. Non-coding. Okay. And so now we're getting further away from the actual coding region here. But we can get a region like this that's called an activator region. So in this particular region here, other proteins can bind that are called activators. So this is an activator protein. So an activator is a protein that often binds away from this area, and it actually requires the DNA to fold. So what would happen is, so we kind of get the basic idea of these names, which are here. The activator could act like one of these signal molecules. The difference is typical signals are not bound to the DNA. They're free in the cell. And we'll talk about the regions of the cell in a second. The activators are actually bound to the DNA itself. And so what would happen is it would cause this DNA to, I'm just going to kind of modify this little drawing here a little bit. We have this protein here, and then we have the activator on this region here. And so we see that the DNA is folding, and then we actually get some action where the activator protein might bind to the repressor protein and remove it, or bind to this region of the promoter and help the RNA polymerase bind. All right, so it can activate a gene to actually turn it on or to upregulate it and to increase the rate of transcription, right, by helping that process go along. So these are some of the basic layout. Now, the other thing I want to mention is that this process here in eukaryotes happens inside the nucleus. But this is going on in the cytoplasm in prokaryotes. So this is all in the cytoplasm for prokaryotes. So for both bacteria and archaea, this is all happening in the cytoplasm. So keep that in mind, because that will affect the transport of the signals, where the signals are made. Sometimes a signal might come from the cytoplasm and then have to be transported into the nucleus in, say, our cells in order to then bind to the DNA. But in bacteria, those signal molecules are produced in the cytoplasm. The DNA is in the cytoplasm, and so it has a fewer steps, and the process could be sped up in that particular way. All right, so this is the first part of what I wanted to talk about, is just to introduce these terms, the concept of activation, the idea of repressible and inducible genes, and then just the overall layout here. So the next thing I'm going to do is talk about operons, and then we'll go on from there. Really quickly, then I'll continue with the operons with this, and I'll erase this while I talk for a minute. Bacterial genomes are small, and they need to be small. Eukaryotic genomes have a large number of intron sequences, the non-coding regions. Okay, so a lot of our chromosomes have these regions that do not code for anything that is actually expressed in the cell. They may be regulatory regions that we've found where some of these molecules can bind, but bacteria don't have. Archaea can have introns, but bacteria don't. Bacteria have this need to conserve space. So having a whole lot of non-coding, untranslated regions, and a whole lot of these regulatory sequences that actually don't directly code for anything is taking up a lot of space in the overall genome. So when you have the genome of a bacteria, bacterial genome, if for every single gene, so if you have a gene here, a gene here, a gene here, gene here, and for every one of those genes you have to have in front of it all these regions. You have to have the operator, and you have to have the promoter, and then you have these other regions, and then you have additional segments that might be activating or regulating in some way. There's going to be a lot of space taken up to these regulation processes. So what many bacteria do is something like this. If these three genes here are all involved in the same metabolic process, so when you studied glycolysis, you have hexokinase and phosphofructokinase and all these different enzymes, they're all needed. The cell needs all those enzymes. So if it's going to run glycolysis at all, it needs all the enzymes that will run glycolysis. There's no reason to express one of them potentially, but not the others. So if you need one, you're really going to need them all. So should you regulate or turn them all on separately or all at once? And so the idea here is that for bacteria, what we have are groupings of genes. So we have a group of genes that in some way work together. And these genes are all regulated by the same operator. So collectively, this group is called an operon. So what will happen here is if we turn on the process of gene expression, we're going to express all these genes, not just one, but we're going to do 1, 2, and 3. All 3 genes will all be turned on at the same time. And so this is one way that bacteria can then save some of their space, right? And not have a whole bunch of unnecessary regulatory regions if they don't actually need them. So some genes might be standalone genes and they're only used for a particular process within the cell, but others that are part of pathways, where you have a number of different genes that are all part step 1, step 2, step 3, they can all be joined together in a way that they're all going to express different proteins, different enzymes, but when they're read, when they're transcribed, they're not done separately, they're done all at once. And the regulation for that process is done all at once. And that particular grouping of them is called an operon. I'm going to go over a specific example of a lac operon, but right now I'm just going to introduce a couple things with terms. You also have things called regulons, which is where you might see even more global control. So maybe you have a different set of genes. So maybe there's gene number 4 is in here, and that's not part of this, but now we have 5, 6, and 7. There are these regulons, which are codes that will then affect numerous operons all at once. So there may be a gene over here, and by activating that particular gene, there are signals sent to both of these at the same time. So the one signal activates several operons. So one operon is a group of like genes. Here we're activating several different groups of genes, but all with a one signal, right? And so this is more of a global signal called a regulon, right? And so again, it's another way of providing certain amounts of control and conserving certain amounts of space within the bacterial cells. So what I'm going to go into next is a little bit about the process of transcription translation and the regulation there. And then we're going to talk about the whole overview of regulation and all the different steps kind of combined, like when and where it can happen, not just at the transcriptional level, but the translational level and other places as well. So how these things can be turned on or turned off, or stopped or slowed down, or sped up at numerous different places and locations.